So, good afternoon to all. I am Dr. Regina, uh, guest faculty in Interdisciplinary Center for Research, Ethics and Protocol, Kochi University of Science and Technology. And my topic is on comparative analysis of proximate and elemental composition of Alitropus typus, coming under Crustacea flabellifera aegidea, exposed to lethal dose of bacterial consortium. And the first I am going to say about the importance of this study. This Alitropus typus, it's an ectoparasite associated with many fish species around the world. And it causes large-scale mortality that leads to economic loss to the aquaculture industry. And this parasite will attach the gills of the normal fish. That is the first figure showing. And the second figure, after attaching this parasite on the fish, the gills will become, that has been shown in the second figure. This parasite is sucking the blood of the fish and that ultimately causing the death of the fish. And so far, chemicals and plants are used to control these parasites, but so many demerits while using these chemicals and plants. And the demerits of chemicals are selection of, by using chemicals in, by using chemicals continuously in the aquaculture farm, it leads to the selection of resistant organisms, then destabilization of beneficial microbes, and it will be deleterious to the environment, and it may even present risk to human health. And nowadays, in many countries, these chemicals are also restricted. And from plants, essential oils have been extracted to control these parasites, but it has also some demerits as it leads to toxic side effects in fish, and it is very difficult to choose the best concentration for each species of fish. And there is always a need to know the sublethal concentration of each essential oil to be treated. And another demerit is its low water solubility, and it has not evaluated the toxic effects on fish communities. And one environmentally friendly approach currently under exploration is the use of microbial communities as biocontrol agent. Biocontrol agent means it is the use of living organism to control the pathogens or diseases caused by them, making it less abundant or less damaging than it would otherwise be. So far, the bacteria, viruses, fungi, fungal and bacterial enzymes, enzyme inhibitors, antibiotics, etc. are coming under biocontrol agent. And my aim was to prepare a bacterial consortium, and studies have shown that this bacterial consortium, it could be more effective than single strains for biocontrolling these parasites. And normally these parasites, its exoskeleton is composed of chitin, protein and lipid, and in which calcium carbonate is deposited. This is the figure shows the exoskeleton of isopod. And in my study, I have already prepared a bacterial consortium comprising of 3 bacteria like Klebsiella pneumonia, Bacillus altitudinus, and Stenotrophomonas maltophilia. And my objective in this study, in this paper, is to study the changes in the proximate and elemental composition associated with the application of developed bacterial consortium on the isopod Alitropus typus. This table shows the proximate analysis of Alitropus typus before and after biological treatment with bacterial consortium, and the values represented are means plus or minus standard deviation of 3 replicates. And the parameters studied are moisture, dry matter, ash, protein, lipid, and chitin. Moisture is the principal component of this isopod, and after treating with this bacterial consortium, its content has been increased to 95.18%, and its ash content has been decreased to 0.57%, whereas its protein content has been increased to 2.85%, and in untreated isopod, it is 2.04%. And lipid also increased to 0.33% from 0.16% in untreated isopod. Chitin, it is the major component in the exoskeleton of this parasite, and it has been decreased to treated isopod, and that indicates the effectiveness of this consortium. As this parasites, its exoskeleton is mainly composed of this chitin, protein, and lipid. And here the moisture has been increased due to, and it may be due to the metabolic activity of the microorganism on the isopod. And the protein has also been increased, and it's also contributed by the bacteria. And the decrease in the chitin shows the ultimate effect of this consortium on the isopod. Next is the concentration of elements in the mid-turgid before and after treating the parasite with bacterial consortium. And this has been done with SEM-EDAX analysis. And the elements present in the untreated isopod, there are 9 trace elements, and the major elements present in the exoskeleton of this parasite are carbon, oxygen, and calcium. And after treating the parasites with this bacterial consortium, The carbon content has been increased to 53.95%, and this may also be due to the presence of the bacteria on the exoskeleton of the parasite. And the calcium content, which is a major component present in the untreated isopod, and it has been decreased to 4.28%. And from this result, it has been proven that the consortium that contains a bacteria known as Klebsiella pneumonia, it has the urease enzyme activity, and that is the reason, and that because of that enzyme, this calcium carbonate has been deleted, which has been present in the exoskeleton. This is the EDAC spectra of distribution of various elements in the mid-turgid of untreated Alitropus typus. Coming to the summary and conclusion, the study indicated that the control of isopod infestation by using this microbial consortium is effective and environmentally safe for use in aquaculture. Differences in the proximate and elemental composition can in turn throw light on understanding the metabolic activities of the microorganisms in treated isopods. However, the precise mechanism and the molecular targets of the consortium against Alitropus typus needs to be further studied. The changes in the mineral content of Alitropus typus may have caused discontinuities in the refractive index of the body, ultimately causing the death of the parasite. Minerals released from the body of Alitropus typus can make an important contribution to the aquatic system or storage in small aquatic species. This consortium also offers an alternative method of supplementing water-soluble calcium, a functional fertilizer to the aquatic system. Developed consortium will make aquaculture operations much easier and environment-friendly. A major outcome of this study is that by constituting this consortium, it is possible to address parasite management in the aquaculture system. And it is envisaged that the bacterial consortium represents one of the most viable strategies to achieve a sustainable aquaculture. And my future perspectives are, next level of validation is envisaged by extensive on-field evaluation of the bacterial consortium for the development of the consortium as a novel parasiticide. The human health and ecological risk associated with field studies in the aquatic environment need to be taken into consideration. As one of the bacteria present in this consortium, that is Klebsiella pneumonia, it is being an opportunistic bacterial pathogen. The future study is also focused on replacing the bacteria with its urease producing results, and this domain has to be further explored to develop the concept into a marketable format. References, these are my references. Thank you.
It is useful, your study will be useful for aquaculture. Another is parasite management.
By killing this bacteria, by killing this parasite using this consortium, we can release the calcium content from this parasite. So it will be a natural fertilizer to the farm.
Thank you, Rajeena, for managing the program, a very interdisciplinary one, enlightening all of us on a very important topic. Thank you very much.
Thank you everyone for giving me an opportunity.